Thanks for the great introduction, Michael. I really appreciate it. Just jumping right in, we're talking about the gut bladder axis today, and this is research that's been in the works for about 10 years ago. Actually, about almost 10 years ago, but give or take a couple months, was the first patient that I enrolled for this clinical sampling collection. So we went through that collection, did the data analysis, and that formed a paper that I'm going to be talking about with you today. It's also a labor of love that came out of a collaboration between the Holmgren lab and the Earl lab, the Holmgren lab here at WashU, and then the Earl lab at the Broad Institute. And the two of the major drivers in that are Colin Warby and Mark Young in the Earl group. So I just wanted to highlight them at the beginning so we could uh, give them all of the kudos that they deserve. So with that, we'll go ahead and move forward. So I don't need to tell everybody on this call how important urinary tract infections are, but just to reiterate the point, there are millions of urinary tract infections that happen each year. There was an article came out recently that the market cap for UTI treatment is going to be about $12 billion in a few years. So by 2030, we're going to be at $12 billion expenditure for treating urinary tract infections. That doesn't include time lost from work and other things like that. And something that also gets lost in this, and in addition to the morbidity and mortality associated with urinary tract infections, just the detriment to the quality of life. People who have frequent urinary tract infections, it's a huge effect on their life, daily life. As somebody who was a caretaker for somebody who has frequent urinary tract infections, it can just be devastating to somebody's general well-being. And then here's where we get to the really scary stuff. About 15% of all of the antibiotic prescriptions in the US go to treat UTI. So a little more than 1 out of every 8 antibiotic script goes to treat a urinary tract infection. So that's depleting our antibiotic arsenal in order to treat this really common disease. And as we know, this is also a disease that's biased towards affecting women, especially women, elderly women, so vulnerable populations. We're going to be talking about ascending urinary tract infections today, which is where the uropathogenic bacteria ascends through the urethra to gain access to the bladder where it causes cystitis, so that inflammation. And from there, it can also ascend into the kidneys to cause pyelonephritis, and then through that to cause sepsis. We're going to focus today on just the lower urinary tract infections, so just the cystitis mostly. And as people know, the majority of urinary tract infections are caused by uropathogenic E. coli, somewhere between 75 to 90% of uncomplicated cases, so cases that don't have another comorbidity, diabetes, catheters, such things like that, are driven by uropathogenic E. coli. And UPEC are becoming multi-drug resistant. So I'm going to leave you with one question that keeps me up at night, which is, what happens when antibiotics don't work anymore? We know that by 2050, more people are going to be dying from multi-drug resistant bacterial infections than from cancer. That's a scary opportunity. So this highlights the need for us to really understand how urinary tract infections come about, and understanding the life cycle of UPEC in order to keep them out of the bladder, or at least keep them from causing disease in the bladder. UPEC are not there's no good genetic definition of UPEC. They're really variable in terms of their genetic relatedness. But what I want to highlight for you here is something that's really interesting, which is that urinary tract infections, which are frequently recurrent, so about 25%, 1 out of 4 to 1 out of 3 of initial UTIs are followed by a subsequent recurrent urinary tract infection within the next few months, 3 to 6 months. What's interesting about that, aside from it being a high number that's a cause for concern, is the fact that about 50% of those recurrent urinary tract infections are caused by the same strain as caused an initial UTI, despite clearance of that pathogen from the bladder. So that, to me, so this is what I'm showing you here. The boxes on this plot are covering instances of same strain recurrent urinary tract infections, where, as you can see, at about half of the cases here, the strain is identical, genetically identical to the causative strain, the initial causing strain. So what this indicates to me and others is that there is some other part of the body, some other reservoir within the body that actually allows for persistence of these UPEC strains through antibiotics, so that even if they're cleared from the bladder, this other extra bladder reservoir is not affected by antibiotics and can then seed subsequent recurrent urinary tract infections. So where could that reservoir be? Well, I'm going to highlight 4 areas that we know E. coli, UPEC specifically, are able to inhabit. We know that they're found in the gut. And we know that they're found in the vagina and in the periurethral area. And it's been known for decades now that the bacteria that reside within the gut often are similar to the bacteria that are in the bladder. And recent advances in sequencing technology have identified that they're actually clonal, that they're the same isogenic strains. What hasn't been well understood is the vagina or periurethral reservoirs as potential reservoir areas. And importantly, also this link between, though we know of this link between the gut and the bladder, it hasn't really been well investigated the same way that we've investigated things like the gut-lung axis or the gut-brain axis, where we're actually able to derive some information about the molecular mechanisms that connect these 2 different body sites in ways that influence disease outcome. And so what I'm going to be, oh, and here, I should have mentioned up ahead, I don't have anything that I need to disclose aside from the fact that I have been playing around a lot with AI generated art. So these different figures that you see here are AI generated, which is why you get this funky little monster over here in the desert that I couldn't pass up. But what's important to note from these areas that I think is interesting about E. coli, and something I want to highlight, is the diversity in the ecosystems that E. coli are able to inhabit. Whereas the gut is this anaerobic environment full of microbial competition, uh, with lots of abundant resources for food, versus the bladder, which has a much lower microbial density, few nutrients, and is a lot harsher. And then in between are the vagina and the periurethral area, which have different levels of oxygen, but also acid stress and all these different things. And yet E. coli is able to colonize in each one of these areas. It's a really fascinating pathogen and its ability to spread throughout the body for these colonization in these in different reservoirs. Um, but back to the topic of our conversation here, we're going to be focusing today on this gut bladder axis. Uh, and for this, we're not only talking about the translocation of uropathogenic strains from the gut reservoir into the bladder. One of the things that I want to broaden the scope of this discussion, not only to the clonality, to the transmission between these 2 body sites, but also the involvement of the microbiome in the gut, as well as differences in host immunity that we have found that we think are influenced by the gut microbiome itself. So what we're looking at is this tripartite interaction between the uropathogens, the microbiome and their reservoirs, as well as the host immunity, and how each one of those pieces fits together and leads to susceptibility to urinary tract infections. So in order to get at this question, the first thing that we wanted to do was to look at the clinical manifestations of this gut bladder axis. Towards that goal, we recruited 28 women, 14 controls and 14 women who had 3 or more urinary tract infections in the past year. And we actually followed them for an entire year. At enrollment, we collected stool, urine, blood, and survey samples from them. And we also collected the stool samples and surveys monthly afterwards. Any person, any subject that developed a urinary tract infection, we would also collect more blood samples from them, a urine sample, as well as stool samples at the time of UTI, as well as 2 and 4 weeks post. And every time they would have another recurrence, we would then go back and track do the same sample collection again. So this gave us a pretty robust collection of fecal samples as well as urine samples at times of UTI and at enrollment, so we could capture some of the population dynamics between those two habitats. And then with the blood samples, we used those for some of the immune measurements that we'll talk about in a bit. And then, of course, with the surveys, we asked about UTI symptomatology, antibiotic use, and both bladder and gut symptoms that might affect the output from this. So the way that I approach science is by asking questions. I really like to start by asking some as fundamental a question as I possibly can, and then moving into a hypothesis and testing from there. And so that's going to be the kind of way that I approach this talk as well. So the basic question that I wanted to start off with is something that we should find. This should be something that we identify, and it's that if gut populations can see these urinary tract infections, and if they do, then the gut and bladder strains should be genetically identical. And so I'm showing you a representative subject that had a UTI. You can see that she has a UTI that's coming from this clade B2 phylogroup, which if you remember, if you had a quick eye, you might have picked up that this is the majority of urinary tract infection causing strains, and that's from the phylogenetic tree that I showed earlier. So most UPECs in the United States come from this B2 and D clades. And so you can see that at the time of UTI, you can see that there was we have one strain in the urine, but in this subject, we were able to capture a variety of different E. coli strains that inhabited their guts at different time points through this. And one thing that we saw from this also were that some of these strains were persistent. So not only were we able to get the phylogroup, we were able to identify them at a strain level resolution. And we could see that there was connections between these time points where the same strain is showing up in the gut, and then you also see this translocation into the urine at the same time as a UTI. And so this is a persistent effect, and what you can see across this, I'll tell this in a bit, is that the majority of the strains that we could find in the urinary tract were actually also found in the gut, so 80% of the time. This is this is at high resolution, so we can actually say that these strains are clonal at the genetic level. And we can do that using a software package developed at the Broad Institute called Strange, which they'd be happy to answer more questions about how to use that really wonderful tool. What's interesting in this subject, and something that we saw frequently, was that even if a person was given antibiotics to treat their recurrent urinary tract infections, you would see clearance of that pathogen from the bladder, but there wouldn't be a reduction of that pathogen in the gut. We would still find that same pathogen that caused the UTI persisting within the gut through the subsequent sample collections. Which indicates that antibiotics were effective in clearing the bladder, but they did nothing for removing the gut reservoir of that same strain. And we think that this is part of a vicious cycle where you're taking antibiotics, you clear the pathogen from the bladder, but you're also disrupting the gut microbiome in ways that don't prevent UPEC from persisting within the gut. And something to note about this is that for the strains that caused urinary tract infections, we saw a lot of persistence in this effect. Again, about 50% of the time after after antibiotics, the E. coli strain that caused a UTI was able to persist within the gut. So now we know that they're clonal. So then another thing that's generally known is that sex is a risk factor for the development of urinary tract infections. And it's thought that this is due to the physical manipulation that occurs during intercourse. And so what we wanted to do again as a sanity check, just to make sure that we were capturing the biologically relevant phenotypes, we looked to see at the sexual history of our control and our UTI women. And what we found was that although there wasn't a significant difference overall, Every woman that developed a urinary tract infection did actually have sexual intercourse in the preceding 2 weeks. So again, we think that this again is something where we're capturing a biologically relevant risk factor, indicating that this physical manipulation that happens during sex is helping to translocate gut E. coli into the bladder. So then once we have this information, we've got a good sanity check. We're able to capture some biologically relevant risk factors and phenotypes. So what we wanted to know then was if there's anything about the gut populations of E. coli themselves that are actually influencing UTI outcome. And so here we hypothesized that these persistent gut colonizers will occur in the bladder more frequently and cause more UTIs. And so towards that goal, again, we collected all of our strains in our RUTI group, and we found, as to be expected, that the number of persistent strains in this B2 or D clades were much higher than in the other clades. And you can see that measured here on this phylogenetic tree on the left, which has been colored by these different phylogroups, B1A, B2, and D. And then next to the B2 and D groups, you can see these large holes here. These are bar plots indicating frequency where we found a particular strain belonging to a sub-branch within the phylogroups. And you can see that the branches in B2 and D are just a lot longer than they are in A and B1, indicating that the number of gut samples that belong to the B2 or D clades are more frequent than what we found in the other clades. And often we found that these strains were persisting between time points, between collections. And then if we add on an analysis of what strains were also found in the urine at those time points, you can see again a preponderance of the strains that are found in the urine at time of UTI belong to these B2D clades, and much less so in the other clades, with only a couple showing up in B1. This is in the RUTI group, but what we were surprised to see was actually something that's basically the same in our control women as well, where we see an increased number of B2D strains within the guts of control women, but we also see that these same B2D strains are showing up in the urines of these control women at more frequently than other clades as well. So overall, what this indicates to us is that the population dynamics, the clade origin, the frequency, and the translocation between the gut and the bladder seems to be pretty similar between these RUTI and control women. Just surprising to us. We were expecting that we would maybe see a shift in the strains of E. coli that were inhabiting the RUTI women versus the control women. We just don't see that. We see that the populations are pretty similar at the genetic level between the two cohorts. So then taking this a step further, we wondered, well, if the strains aren't different, maybe there's just more of them. Maybe the strains that are in the gut of RUTI women are at a higher abundance. And again, we saw that this wasn't the case. We saw this on the top here, showing just a bar plot showing the relative abundance in the gut microbiome. And this is based off of whole metagenomic sequencing. And you can see that in both the RUTI and control cohort, somewhere between 40 and 50% of the time, we weren't able to detect any E. coli in the whole metagenomic samples. And if you take away that chunk of the population and just look at the samples that had detectable E. coli abundance, you can see a similar score between them, a similar amount of burden in terms of relative abundance between the RUTI and control women. So there doesn't seem to be overall a difference between not only the types of E. coli that are in the guts of RUTI versus control women. No, we also don't see a change in the relative abundance between RUTI and control women. The next thing that we wanted to look at is, well, maybe there isn't an effect overall, but what about at time of UTI? And so what we did was to look at samples both preceding and after and following a urinary tract infection. And what you can see in the bottom here is that in general, there wasn't a strong trend with an increased amount of E. coli preceding a urinary tract infection within the gut. Now, we don't have a lot of temporal resolution in this. So what that means is we were taking monthly fecal samples, and sometimes women would have a UTI mid-month, or they would have it, sometime, weeks after they had provided their previous fecal sample. So it's possible that we don't have the type of resolution where a bloom may happen within days preceding a UTI. But overall, what we can say is that there doesn't seem to be a significant buildup of E. coli in the guts of these women for months before they have a UTI. We just don't see a statistically significant effect toward that would support that type of analysis. Well, okay, so we've looked at the E. coli population dynamics, but of course, E. coli aren't the only bacteria that live in the gut. So if there isn't a difference in the E. coli between RUTI and control women, what do we think will happen if we look at the rest of the microbiota? And here we found that there was a significant difference here. And looking at microbial richness, so looking at a measure of the differences in abundance of distinct species within the gut microbiota, we saw that there was a statistically significant reduction in the diversity of the gut microbiota of RUTI women relative to control women. And this is important because in general, a greater diversity of gut microbiota is associated with better health. And a reduction in the diversity of gut E. coli is generally associated with something called dysbiosis and generally associated with poor health or poor gut health overall, poor community health. We've talked a lot about antibiotics here. And of course, these RUTI women are taking a lot of antibiotics. And so is that the driver of this difference in the microbial diversity in the gut? And that doesn't appear to be the case. So what you can see here are two bar plots. On the right-hand side, you can see that microbial richness metric that we talked about already. And on the left, you can see these frequency bar plots indicating how many UTIs different women had, as well as how often they were prescribed antibiotics or how often they self-reported taking antibiotics. And you can see that the RUTI group did consume more antibiotics than the control group. But what we don't see is that women that took a significant amount of antibiotics sometimes had reduced microbial richness, but other times there were subjects that took lots of antibiotics that ended up having relatively high rates of microbial richness in their samples. And so it doesn't appear to be that these antibiotics are strictly the reason for this reduced diversity. There's not enough of a correlation between antibiotic use and reduced microbial diversity within these patient samples. So that's interesting because that does indicate that maybe that there's some persistent effect that's happened within these RUTI women that's keeping the community in this dysbiotic state. And so delving into that a little bit more, not only did we want to look at species counts and frequency, we also wanted to look at the composition. So what bacteria are actually there? What's their identity? And we found that if you look, you can pick out a few of these higher taxa, like Bacteroideaceae and Ruminococcacea, that are significantly different, where we see an increase in the Bacteroideaceae in the RUTI groups, and then a loss of the Ruminococcacea in the RUTI group relative to controls. And so there are some things that we saw in terms of these higher taxonomic levels. When we get down to the species level, there really wasn't much that we were able to capture, and that's probably due to the low number of patients or subjects that we had in the study, just 14 between the two. And then once you do we didn't have the power to really pick up the differences between these two. But one thing that we did see that was striking to us was a difference in these levels of these butyrate producers. So butyrate is a short chain fatty acid, or SCFA for short. And these short chain fatty acids have a lot of roles in human health. Butyrate is generally considered to be anti-inflammatory, but it's it is absolutely immunomodulatory. Butyrate is part of a signaling network that microbes provide to the host that influences the immune tone. And so one thing that's interesting about this is we see this reduction in butyrate producers. And you can see again, if you go through, you see differences in a few different microbes. But something that you may have seen in other studies is this F. prausnitzii that's been associated with IBD and also with Parkinson's disease and quite a few different inflammatory disorders that have been connected to the gut. And so within this RUTI group, you're seeing a reduction in the microbes that are known butyrate producers. That doesn't necessarily mean that you're seeing less butyrate in the samples. So we took it a step further and actually did a metabolomic quantification of the amount of butyrate within the fecal samples of our different cohorts. And we found that matching our previous results, that there was a significant reduction in the butyrate levels within the feces of these women. So not only are there fewer butyrate producers, we're actually capturing a reduced level of butyrate within these samples. This is, again, I brought up IBD, but this is interesting because butyrate has been so strongly associated as being something that helps ameliorate the inflammatory pathology associated with inflammatory bowel disease. And so to understand this connection a little bit better, we did a correlation analysis where we took the microbes within our study, from the RUTI cohort, and then did a correlation against the samples that are associated with IBD guts. And what you can see is that there is a pretty strong correlation between the abundance of the bacteria in our group and the abundance of that same bacteria in an IBD subject. So that you can see that in general, that the circles are similarly sized and similarly placed on this graph. There are some differences. In general, IBD sees a reduction in Bacteroides species, and we saw a trend towards increased Bacteroideaceae. So it's not a one-to-one comparison, but we didn't find anything that was lower in RUTI and higher in IBD, for instance. So there's a general similarity between these two diseases. And I want to note that we did not find any evidence of anybody in our cohort, our RUTI cohort, as having IBD. We actually specifically screened and used that as an exclusion criteria to make sure that people with IBD weren't coming into the study. But we also asked about diarrhea, gastrointestinal pain, things like that, but we didn't see this. So what we're thinking is that there's some similarities, but it's not a one-to-one comparison where these RUTI women have IBD-like disease. And again, I'm just putting this in a little bit nicer. We see the similarities between IBD and this RUTI gut microbiomes are found in reduced short-chain fatty acids, reduced diversity, and reduction in these specific butyrate producers. So this is the general architecture that we found. We've seen that the gut microbiome of these women with recurrent urinary tract infections are dysbiotic and have distinct composition and metabolomic profiles, right? We're able to pull out some of that information. But what does that mean for that other part of that triforce that I showed you at the beginning? We're looking at that immunity. And here we found more mild phenotypes, but still statistically significant effects that are intriguing. One of them is that looking in the serum of these women, we found that at enrollment, that control women had a reduced amount of this eotaxin cytokine. Eotaxin, otherwise known as CCL11, is associated with eosinophil recruitment and has been associated with IBD, but also enterogastritis, and a few different diseases where you're seeing this inflammatory response that brings in eosinophils that causes damage to the surrounding tissue. Intriguingly, eotaxin also happens to be upregulated during a UTI, significantly upregulated during UTI. And then if we were to take out some of the immune cells from the blood and just look at their RNA gene expression, we saw that there was a significant uptick in these two genes here that don't have a strict that are associated with pro-inflammatory states within PBMCs. And what's interesting is that this, I think it's this link 00944 has also been picked up as being associated with bladder inflammation in overactive bladder. So of all the things to pick up, it was interesting that that was the one thing that we picked up in sort of this systemic immune system profile. So what this is leading us to believe is that there might be some sort of mild change in the immune tone of women with recurrent urinary tract infections, but we're not seeing this kind of over exuberant IBD-like constantly feeling bad profile that happens in other inflammatory diseases. And that actually kind of makes sense because women with recurrent urinary tract infections aren't displaying the same signals of a systemic pro-inflammatory signaling cascade that others might have. So just to summarize this first part really quickly, what I'm going to talk about is the fact that we are able to find that sex plus UPEC in the bladder was associated with a UTI. We found that UPEC in the gut are clonal with the UPEC that are in the bladder. But what we also found that antibiotics were effective at clearing the pathogen from the bladder, but ineffective at clearing the UPEC from the gut. It was basically a coin flip as to whether or not that was actually happening. And then we also see evidence of this gut dysbiosis and its connection with an immune response that we're attempting to characterize further. We're interested in looking at the immune response within the gut microbiome itself, within the gut itself, in mouse models to see if we can recapitulate this phenotype. And then something that we don't know and we're working on, we have some preliminary data that it's a little too early to share, that these immune responses that we're seeing are actually having an impact on UTI outcome within a mouse model. Though what's striking to me is something that I mentioned earlier, something that we didn't see, was that the UPEC in the gut, I was really expecting that we would see some sort of bloom, increased abundance, or some change in strain identity in these recurrent urinary tract infections in these women. And I was more sure of that when we got the data that the gut, despite that we saw these levels of gut dysbiosis. So the fact that we didn't see that was really intriguing to me. So I wanted to take we wanted to take a step to try and understand if there was a connection between UPEC in the gut and this gut dysbiosis. So if there isn't a change in the population dynamics, maybe there's a change in their lifestyle. Maybe they're adopting a more virulent profile in the gut, and that's what's driving this increased bladder colonization phenotype. And so that's what we looked out to try and get. But the problem with this is, as we mentioned before, that E. coli is just such a low abundance in the gut microbiome of both RUTI as well as healthy women. And then but there is a way to get around that, and that's something that was developed with help at the Broad Institute to use this selective amplification of reads that were then sent for RNA or DNA sequencing using biotinylated probe set. And so what these geniuses at the Broad Institute did was to create this modular tool called E. coli PanSelect, where I won't get into the details with this, but they were able to select hundreds of thousands of these nucleic acid probes that are able to bind to either DNA or RNA, and where they're able to collect the DNA and RNA in a complex mixture and pull them down and increase the sequencing efficiency of these of E. coli by enormous amounts. And so to get into that a little bit more, what they were able to do is we together actually tested a mock community composed of 4 different E. coli strains. And what you can see is important on the left, you want to look at the y-axis. You can see in that before this hybrid selection procedure, before HS hybrid selection, you can see that we had a collection of 4 E. coli strains that made up 1% of a mixture. The other 99% was random human DNA. Did you guys know that you could buy, human DNA and they just send you like a powder of it, like a protein shake. They send you human DNA. It's really cheap. But then what we're at pre-hybrid selection, it's at 1% abundance. But after hybrid selection, you can see that that jumps to over 30% of the relative abundance of the sequencing reads move from being that move to E. coli strains. So you can see that on this little line chart here, you can see that there's a significant increase in all of the strains, and that leads to an increase in the depth of coverage across the different strains, and that importantly, that the relative abundance of each strain within the community did not significantly change. So that this H10407 is at the same relative abundance to other E. coli pre-hybrid selection as to post-hybrid selection. So there wasn't a bias in some strains being covered and having more sequencing reads than others. So this indicates that it's unbiased amplification of a mock E. coli community, and 30-fold at the DNA level. And what's striking about this is you can also apply this to RNA, and in both cases, you can take samples where E. coli is not undetected at both the DNA and RNA level, and then after this hybrid selection, you can see the significant amplification of these things from non-detected, which would be underneath this line here, all the way up to these bits where you're actually able to get the sequencing information from both DNA to get the strain level identification, but also RNA to look at the gene expression of these strains within this complex community. And so I'm not going to go into more detail about this, but you're also able to use this as input to look at metagenomics, but also metatranscriptomics. So this is a really wonderful tool. And anybody that's interested in doing this type of analysis, I would strongly suggest reaching out to Ashley Earle about this wonderful system that we've developed together. Now, back to our question at hand. Is there a difference in E. coli lifestyle? And what we found looking at E. coli that were in the recurrent urinary tract infection women versus the control women, we saw that there was a difference in the expression of a limited number of genes. So only a few individual genes by themselves reach this level of statistical significance, which is surprising. There is a great diversity in terms of the genetics between E. coli strains, but still, it was wonderful that we were able to see some differences within this bit. But what was really wonderful is when we took all of these genes and collapsed them into their different regulons. So looking at the transcription factors that actually determine their relative expression, you could see that a couple of things popped out really quickly, and that related to these global transcription factor regulators, FNR and ARC-A. So I'm going to be talking a lot about this FNR and ARC-A in a little bit, but for right now, just know that they are involved in the transition of E. coli from this fermentative anaerobic respiration to aerobic respiration, where they're using oxygen as a terminal electron acceptor. And that's actually associated with growth when there's oxygen in the environment, whereas when there's no oxygen, you have this anaerobic growth profile. FNR is both a repressor and an activator. So it activates anaerobic respiration machinery and represses aerobic respiration machinery, while as ARC-A is a repressor. So it really only represses aerobic machinery. So what you're seeing here is that genes that are normally repressed by ARC-A, so these ARC-A genes that are normally repressed, which are aerobic machinery, are actually significantly upregulated within this RUTI group. So what that indicates to us is that in an RUTI group, there is a de-repression by ARC-A. ARC-A isn't doing its job as well as if it was in a control person. And you see a similar thing with FNR, where the genes that are normally activated by FNR are actually significantly lower, have lower levels of expression, meaning that FNR isn't activating as much. So take that together. It indicates that these strains appear to be experiencing a higher or responding as if they are in an environment with increased aerobic respiration potential. This also extends to this nitrate-based respiration, where instead of using oxygen, E. coli are able to use nitrogen and nitrogen metabolites as electron acceptors. What you see again is that there's this change where E. coli that are living within these RUTI gut microbiomes appear to be experiencing increased oxygen, and they also appear to be responding to an increased availability of these nitrate electron receptors. I'm not going to give you the whole spiel, but we also see additional differences in energy and biosynthesis pathways associated with ribosome construction and a few different carbon source utilizations, including fumarate and some things like that, and also fumarate metabolism. But in general, the strongest phenotypes that we saw coming out of this was this association with an increased aerobic and nitrate-based respiration relative to control women. And as I mentioned at the beginning, one of the important things about the gut is that in general, in a healthy environment, the gut is anaerobic. It has very low oxygen concentration in the colon, where E. coli is at its highest burden. So this is a striking result that we're looking to follow up more on in the future. To put that into this graphical abstract form, this is what we've put together. Is that you see these FNR regulated is generally down regulated in UTI. And so you see a reduction in fermentation and this fumarate respiration. But you also see this increase in this ARC-A regulated respiratory metabolism. So ARC-A normally represses these genes, but in RUTI women, they're increased expression relative to control. And that includes both oxygen as well as nitrate-based respiration. And that could be, we don't know where this is coming from. We don't know if there's increased oxygen or increased reactive oxygen species or reactive nitrogen species. We're currently investigating that in some mouse models, but this does paint a picture where this RUTI gut microbiome is living in a place where there's an inflamed epithelium, where there's mild inflammation. This is concordant with a model where E. coli in these RUTI women are existing in a mild inflammatory host. And so we're really excited about this because we think that this may help explain why we're seeing this difference in this gut microbiome composition. And we're looking to see if this mild intestinal inflammation might also be predisposing to a altered UTI outcome. And so that's all the information that we put together. I'm going to give you three major takeaways that we've got from this gut bladder axis, which is that the gut populations of UPEC are clonal and that it persists through antibiotics. That the microbiome in RUTI women appears to be dysbiotic, and a significant signal driving that fact is this loss of butyrate producing organisms. And that we do see evidence that there is changes in systemic inflammation with this eotaxin, but also this we are seeing signals that E. coli are seeing mild inflammation in the gut. And so with all of that together, there's lots of people to thank for all of the work that I presented today. Lots of people in the Holgren lab, the Cal lab, and the LA lab, all at WashU, and then at the Broad Institute, a murderer's row of bioinformaticians that helped put all of this information together for us. And with that, I'd love to, of course, funding sources, that wouldn't be possible without money. And so with that, I'd love to take any questions that anybody has.